Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Spector.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in creating a better tomorrow. Today, we have the honor of being joined by Dr. Ira Spector, co-founder of SFA Therapeutics, which is a microbiome-derived biopharma company located here in the Philadelphia area, focused on really new advancements in the treatment of a range of inflammatory disorders. Dr. Spector is a very experienced drug developer, has over 3 decades of experience, and has have developed dozens of approved drugs, including big names like Enbrel, Effexor, Protonix, Prevnar 13, Mylotarg at Wyeth and Pfizer. There he served as vice president of clinical operations and vice chief of development. He was also at Allergan, where he helped develop another range of products, Ozurdex for diabetic macular edema and retinal venous occlusion, was involved in development of Botox for a variety of other indications, including overactive bladder, chronic migraine, even in the area of cerebral palsy. Prior to Wyeth, he was VP and partner at the PA Consulting Group, and then before founding SFA, was the executive vice president of analytics and consulting at the CRO Icon. Dr. Spector holds bachelor's degrees in physics and electrical engineering from Washington University, went on to do an MBA at Drexel, and then did his PhD in health sciences at UMDNJ, Rutgers University. We're honored to have him with us. A lot of really interesting themes to get into today. Dr. Ira Spector, thank you so much for taking time to come on the show.
Thank you, Ira.
It's great having you. I would really like to start off a little bit with your background, because I find it fascinating. You're studying physics and electrical engineering, and you go ahead very quickly into the pharmaceutical industry, and not just a lot of the early stage discovery stuff, which we'll be getting into in a little bit, but some of that later stage drug development, clinical development we don't normally discuss as much on this show, but would love to hear a little bit about your background and a little bit about what got you into pharma development in the first place. If we can start off there.
Okay, so I created essentially a biomedical engineering degree before there was such a thing, by combining physics, electrical engineering, and I also took a heavy load of biology. I worked in medical devices for a fair amount of time. Actually was infamously famous for a consulting project that we did for J&J Ethicon, where we put a camera chip on the end of a flexible endoscope. And so anybody who's had a colonoscopy, that was the technology that we helped them develop. I did that for about a decade, working on a number of devices, and my background in physics taught me how to model processes. So it's really about first principles. How do you model a process? Engineering teaches you how to apply processes. When I had an epiphany around in the late 80s that all the pharma companies were buying device companies, and I'd better learn something about drug development. And I decided to do that by going to work for a consulting firm. PA Consulting is an international strategy consulting firm. And I built a practice around R&D strategy and speed to market, and worked for most of the big device companies at the time. helping to write strategic plans for them, helping to analyze projects and businesses. And then got a phone call from a headhunter who said, there's this company that makes Echo pots and pans, O-Cedar mops, Duplicolor car paint, Jiffy Pop, Chef Boyardee, and oh yeah, they own 3 drug companies and they want to become a drug company. And that was American Home Products.
Yep.
And they're looking for a head of strategy, and we'll give you a clean sheet of paper to start a drug company. And we're bringing in an esteemed new head of R&D, and that was Bob Levy, who had been one of the fathers of the Framingham study that linked cholesterol to heart disease, and at the NIH at the Heart Lung Blood Institute, and had come from being head of the med school at Tufts and then Sandoz, head of research. So can you come in as the internal consultant and help build a drug company? You cannot turn those kinds of opportunities down. I left PA, where I was a partner running their US healthcare practice, took a demotion to join American Home Products, and over the next 17 years, we built Wyeth. My first observation when I joined Wyeth was the MDs know everything they could possibly know about the biology, but very little about process efficiency.
Yeah.
Engineering teaches us how to be efficient with processes. And in fairly short order, I got drafted, or it somehow became apparent that I knew a little bit about business processes. And I had been consulting in business process reengineering anyway. And I got drafted to develop a system for tracking all of the clinical projects in the company. And we implemented that system called Impact. Then it became apparent that I was good at managing things, so I wound up responsible for clinical project management. And then I got a new boss, Bob Ruffalo, who on my first day I said, I'm responsible for, he said, what do you do? I said, I'm responsible for every date and deadline that your organization has to meet, but I have no control over the processes and they're not very good. So he turned around and said, fine, you're now in charge of fixing them. And I wound up building a fairly large cross-functional team to re-engineer clinical operations from a clean sheet of paper all over again. And we built Wyeth clinical operations starting with 8 people. We became 1,750 people plus 700 contractors. We outsourced data management, moved data management to India. We borrowed optical scanning technology that was being used by the credit card companies to read invoices, to read the paper. Case report forms, we went completely paperless. By 2007, we were 99% paperless. We built our own electronic data capture system before there were such things. We pioneered modern clinical operations and created the systems and technologies for managing. At our peak, we were running almost 500 clinical trials at a time, with over 50,000 patients recruited at a time in 67 countries. So the engineering and physics background fit very well in a clinical environment because it enabled me to see things differently than the traditional way of learning the ranks or the ropes in clinical development was usually an apprenticeship. You learn from someone else and you did it the way they taught you. It enabled us to break a lot of molds and change a lot of processes. And I like to think we were one of the first to do that in the industry. Now, a lot of what we did is standard.
It's interesting thinking about what you're saying here. And again, I read in your bio that you were involved in novel indications, small molecules, biologics. You did repurposing, and then I noticed along the way, it's what you did your PhD. It's sort of in the middle of sort of, I think, when you were at Allergan, predicting clinical research site performance, accelerating development of healthcare therapies. And I found this so fascinating because it's again one of those areas that we don't think as I remember paying CROs in the past in former careers, and okay, they're in charge of that, and I don't have to worry about it. But no, there's a lot that goes on in that period, even though we're well past the valley of death. that used to concern us, to actually get proper clinical work done. And I just love, a couple minutes of, sort of top line learnings from that era, Ira, as we get into what you're doing now. But just, here we are in 2024, key takeaways from efficiencies in clinical in clinical performance, clinical research site performance. Some takeaways from that period, if you would.
So here's the fundamental problem. How do you know which site is going to enroll patients?
Right.
And that is still the fundamental problem in this industry. And it is, it is why the maxim of the industry for years has been, in real estate, it's location, location. In clinical research, it's sites and sites and sites.
Right.
But how do you know which sites are going to enroll? And the problem, the problem has been so bad that we always assume that sites will overpromise by 50% and under deliver by many months, and that's the maxim of the industry. My thesis analyzed 18 attributes associated with site performance, and I did find an indicator actually that links very well to site performance. And the other interesting thing was enrollment performance also links to site quality. So sites that tend to enroll well tend to have fewer queries and less unresolved queries. So what I was looking for is how to create predictive analytics that we could use in site selection to weed out the poor sites and pre-select the better performing sites so that we can enrich the trial performance and hit our performance metrics as an organization. And we employed these techniques at Wyeth, we employed these techniques at Allergan, and we found that they were pretty effective. Clearly at SFA, we're doing some of the same work. My thesis is public, but to date, I haven't seen anyone deploy these in a large way, and maybe that's another business or career, but clearly there are predictive analytics associated with site performance and patient quality and data quality. And rather than brute force, we're just going to spend a lot of money on a lot of sites. We all have budget constraints, and we built clinical operations as a machine that could get more efficient every year, and by cutting cost and time. And this was one of the tools that we deployed.
So let's transfer now to a little bit of what you're doing now. And before we get into SFA, the company, would love to talk a little bit about sort of the background and the research of your co-founder, Dr. Fedelson, who's down the street here at Temple, that's been working, had an extensive career in looking at microbial metabolites, the relation to health and disease. And, this is an area that, obviously we have a pharmaceutical industry throughout the 20th century, which was extensively based on, small molecule skeletons that came from the microbial kingdom. The human microbiome, a little different story, just because we have these organisms that live in us, on us, symbiotically as a part of us, that, some are not all bad, do a lot of good things, have to hang out, protect us from bad microbes, have to do interesting things in terms of the biology to make sure that we don't kill them, and so forth and so on. A really interesting little ecosystem. And as you're Again, Dr. Fedelson, is a recent paper entitled Microbiome, Metabolites, and Health and Disease. This was just in September of 2023 in Frontiers of Microbiology. Talks about sort of the broad extent to which this microbiota is in every organ and tissue throughout our body, and really does amazing things to keep us healthy and well. Talk a little bit about his background, if you would, and a little bit about sort of the genesis of some of the concepts that lead to the formation of SFA.
So let me start with Mark. So Mark did his PhD at UCLA Geffen Med School and his postdoc at Stanford. Came to the East Coast to the Fox Chase Cancer Center, also Philadelphia based,
Yep.
To work for Barry Blumberg Barry Blumberg won the Nobel Prize for discovering hepatitis B.
He was my neighbor for.
So,
I could throw a rock at his house.
Okay, Mark continued that research at Jefferson and then at Temple. And his fundamental work has been looking for a way to block the inflammatory mechanisms that lead to fibrosis. Fibrosis leads to cirrhosis. Cirrhosis leads to, a liver cancer. Actually, it's hepatitis, inflammation, and then fibrosis, cirrhosis, and that's the cascade in liver cancer. So Mark was looking for a way to block that mechanism, and he realized there was an analog in the microbiome in another disease, and he asked whether or not he could transfer that finding to the liver. And Mark had colonies of mice that have the HBX site. They develop liver cancer, left untreated. HBX virus leads to liver cancer. They all get liver cancer and they die. And Mark did some experiments and found that if you treated adult mice, half of them don't get tumors, the other half get very small tumors, and those tumors, the liver regenerates, so they never really evidence as being a problem. And that was a breakthrough. But I think what Mark really did was he looked at the microbiome and he asked a fundamental scientific question, which I don't think many have asked. Instead of focusing on prebiotics, which mostly do nothing because they don't get to the receptors where they're effective, really the bacteria themselves, which are, that's personalized medicine because your microbiome and my microbiome are probably different. And I've gone to talks where they do fecal transplant, which is what it sounds like, and they transfect and you take a sequence from patient A and a sequence from patient B and you get Q. I mean, it's not very reproducible. Mark said, well, what do the bugs make? And what's their physiological function in the human body? They must be there for a reason. And what do those substances make? And if we can make biosynthetics of those substances and figure out what those substances do, perhaps we have a whole new class of medicine. And so that's Mark's fundamental question and discovery. Our other co-founder, so Mark is the F in SFA, I am the S. Our other co-founder, Ira Spector, asked, well, how can we make those translatable into something to dose into humans? So she became our translational scientist. And my contribution was I looked at the PCR microarray data and I was able to pick off targets and match them to diseases and said, if you can increase the selectivity against specific diseases, we can turn these into drugs. And that's what Ira did. So there are three primary inventions involved: recognizing the targets, understanding that these substances have a fundamental physiological effect in the body, and then figuring out how to dose them and how to create sustained blood levels. Because the other thing about this class of substances made by bacteria, in this case in the GI microbiome, although there is a microbiome wherever the human body reacts with the exterior world. Yep. These substances have potent effects in modulating the immune system, but they have exquisitely short half-lives. So being able to drug them and turn them into biosynthetic drugs is part of what Ira contributed. So it was really the three of us working in the lab at whiteboards, having many, many discussions and thousands of hours of arguments that kind of led to the genesis of this company.
And you mentioned immunomodulation. So obviously you have these really fascinating mechanisms. You got to make proper druggable systems for humans. Could you say a few? Because I noticed, in the literature, and I saw one of your corporate presentations, this one, the lead asset known as SFA 002, does quite a bit of, interleukin 17, 23, TNF alpha. So really, shows that there's a broad spectrum of sort of evolutionary perfected capability here. But then I noticed something that said, and this was equally fascinating to me, that is SFA, can you say, it looks like it's a combination, though, of these fatty acid moieties.
Yes.
Okay, because that is extremely important here. I wanted to sort of put that on the table as well, if you could talk to it, because the concept of combinations, has come back in a major way, obviously, after sort of the HIV story that we might not have to combine drugs that are already approved, but start with combinations. And in these particular cases where these microbes make all sorts of interesting bioactive substances. They don't make single drugs like we do, they make these interesting combinations to deal with their environments. Can you talk about that as well? Because that's another really important aspect of having someone with clinical experience, how you push a gamish, let's say, something that's not exactly a purified entity into development.
So after 40 years in the industry, the industry mantra has always been single channel, single target.
Right.
And why single channel, single target? Because of control of safety.
Right.
Because the concern is if you have off-target effects, you could have off-target safety issues, and of course, that's the way the industry has approached drug development for very many years. The human body is not designed that way. As Mark Feidelson has said many times, the human body was created with many parallel paths, and upregulation and downregulation of those paths leads to a homeostasis of managing various physiological functions. And that's what these substances do. I'm very quick to point out that some of the most successful drugs out there are pleiotropic and hit multiple targets. My favorite example, of those is metformin, which is still not fully elucidated. Aspirin does some of the same things. And having worked in drug development, I can remember when we developed Effexor, and we thought, wow, we have a two-channel approach to depression, right? So an SNRI, and we're going to hit dopamine, and we're going to hit norepinephrine, and we're going to hit two channels simultaneously. And isn't that great? And it probably worked in 40% of patients. It turns out there's multiple channels in the human body. What's interesting about biosynthetics, that are drugs that are made to mimic natural substances found in the body, is that those natural substances tend to hit multiple targets. And so what we're doing is synthetically making the same substance with the idea of hitting multiple channels. Now, why have a polypharmacy approach or a multiple fixed dose combination? Because I'll go back to the very first drug, which was SFA 001 that Mark discovered in his lab at 12 Denaris Street, which was for liver cancer, for which we have 2 patents and orphan drug designation. The problem is that if you look at the channels being downregulated and you do the PCR microarray-analysis, there are multiple channels. And I exactly asked that question of Mark back in 2016. I said, can you make this more selective so that we can focus on one of these channels and not all of these channels? And the solution was to add additional substances, much like you do in vaccines where you use adjuvants to increase selectivity or potency. We have done a similar thing here. We also call them adjuvants, but we're using these substances differently. And Alla came up with that, I think, very brilliant insight. And that's enabled us to shift where we're focusing the effect. So we're not working with all short chain fatty acids. We're working with selected ones with selected adjuvants in a selected dosing regime to target specific targets in the body. And that's how we've developed 8 drugs. 002 being our second drug, which has been optimized for autoimmune disease and for which we've patented it for I believe it's over 40 different conditions, but we focused on psoriasis as the initial gateway to autoimmune disease. But the use of these adjuvants does make it polypharmacy, but it also increases selectivity, and that's what we were looking for. So this is a kind of rational drug development applied to natural products made biosynthetically.
No, I'm excited. It's extremely elegant, and having had my having got my foot wet in combination development back in the 90s, I root you on because I think it's definitely the path towards addressing many of these multifactorial sort of unmet medical needs. So I'm excited for you.
Thank you.
Talk about psoriasis. Talk about your phase 1A a proof of concept study, and then a little bit of what you're planning for the next part of that, because I know you're recruiting, I think, currently for a larger psoriasis study.
So before I agreed to form a company, I asked for two things. I said to Mark and Alla, who I met at the lab, first, prove this works in a human model, because remember, Mark had very elegant experiments in mice. And second, show me that you can make this more selective. And they accomplished both of those and showed me the data, and they ran a small study outside the US in a handful of patients, and the data were spectacular, at which point I said, okay, let's form a company. So that's was the early genesis. Now, initially, we were looking at liver disease. The problem with liver disease is prove that you prevent a disease by giving a drug. First of all, the disease is very slow growing. It has about a 10-year to 20-year progression. And secondly, proof of prevention is a very difficult trial to run.
Sure.
So we pivoted to another disease, and psoriasis being the disease we chose. But we're not per se just a derm company or just a psoriasis company. When you go to prove that a drug works in a disease, you need a good biomarker to show efficacy. In derm or in psoriasis, the skin is the most elegant biomarker you can think of. So instead of having to develop a sophisticated biomarker, you see it in an organ. You see it on the surface in skin. The other thing about psoriasis is it has a very high incidence of comorbidities. Patients with psoriasis often develop psoriatic arthritis. Something like 80% will progress to psoriatic arthritis. Patients with psoriasis often develop RA or have other comorbidities, RA, lupus, etc. And they almost always have GI issues. IBS, IBD, Crohn's, etc. are highly correlated. Now, in Western medicine, we treat each of these as separate and distinct diseases. So you see a rheumatologist for rheumatoid arthritis, you see a dermatologist for psoriasis. Sometimes those are treated together today with agents like Enbrel and monoclonals, but you'll see a GI doc for IBS, Crohn's, etc. What if they all have a common root cause? What if they're all related to a defect in the GI microbiome and the inability to make these substances endogenously? And what if we can provide these substances as a drug or as a prodrug and replace what the body isn't able to make in these patients? That's the fundamental hypothesis behind SFA. Just like you give insulin, or we give insulin to type 1 diabetics to replace what they can't make in their pancreas, we can give these substances to patients who aren't making them in their GI. And in the phase 1A, we had 2 subjects who we treated for long duration. Their psoriasis cleared 100%, and then we withdrew the drug. And 2.5 years later, they are still symptom-free. So they've had a complete response. Now, our working hypothesis is that their GI microbiome was remodeled, and they were able to make these substances on their own. We won't see this in phase 1B because the treatment duration is too short. Treatment duration is 3 months, initially, and then there's a 3-month extension that the FDA granted us to allow patients to continue on therapy. But we have seen 100% responses, and we have seen patients continue to improve even after the end of the trial, and continue to improve even after the 3 months into the extension for 3 months. So that tells us that something is happening, and that's very exciting. As we design phase 2, we're going to be looking for longer duration treatment effects. But what we're really talking about is, can we find a root cause for autoimmune diseases based on the inability of making these substances and providing them exogenously, rather, to enable patients to then remodel. And that's what we're doing with our biosynthetic drugs.
And as you point out, that remodeling can, as autoimmune is such a wide swath of conditions. I mean that there's just so much potential there. I mean, obviously, psoriasis is, as you were pointing out, here's the key biomarker. We can address that, but the ability for sort of the trickle down to everything on that autoimmune list, which I forget how many Americans have some form of autoimmune nowadays. I think something like 70 million. So I mean, you're clearly on a path there with sort of a platform that can address a range of conditions in that category. Can you say a few words? Because I noticed that you, Dr. Fatal and Ali, you were all in this paper recently entitled "Short Chain Fatty Acids and Cancer Pathogenesis." And this paper, this is September 2023, goes into some of what you're talking about in terms of homeostasis and dysbiosis of the microbiome, but then it gets into something equally interesting on basically how the microbiome potentially is also affecting, and this is a very hot area nowadays, needless to say, the epigenome. And
Right.
again, when you're dealing with these different sort of early stage genes, the Wnt pathways and hippo and hedgehog and all that stuff, sort of the early developmental stage that leads to development, but also cancer. Again, the ability to play in that space is extremely hot. I'd just love for you to say a few words, if you could, about some of
So
what you envision in oncology.
there are a number of cancers that develop on an inflammatory background.
Right.
Patients who have pancreatitis, which is often undiagnosed or underdiagnosed, but patients who have pancreatitis have a reasonable potential to develop pancreatic cancer.
Yeah.
Patients who have hepatitis have a reasonably high progression to hepatocellular carcinoma, the most prevalent form of liver cancer. These itises, or inflammatory conditions, are often a backdrop in the epigenetics of the development of tumors. And what the short chain fatty acids do, we believe, is downregulate that inflammation. And so the concept here is, can we alter the course of epigenetics by altering the inflammatory mechanisms using these substances? And if you look at the prevalence of various short chain fatty acids in healthy adults versus the prevalence in patients with these underlying conditions, there are striking differences. And so the question is, can we change that? And what's really exciting is these substances have evolved in humans for eons.
Yep.
So they lack innate genotoxicity as long as the doses are controlled and managed to levels that approximate what are found in healthy humans, which means that they ought to provide a much more safer approach for treating these underlying background inflammatory conditions. We're also speculating that they may have an effect in the tumor microenvironment. We're doing some very basic research in our lab and in a sister lab with another university, where we've put an agreement in place to see if we can alter tumor microenvironment. And in doing so, perhaps make certain forms of cancer more amenable to standard of care with lower doses. Because what makes cytotoxic treatment difficult is the side effects associated with them. And so if we can alter the tumor microenvironment by tamping down the inflammation and making those tumors more susceptible to treatment with cytotoxic agents, perhaps we can lower the dose of those agents and have a much more effective therapy. So we are looking at some adjunctive approaches, but we think this is fundamental science that has largely been overlooked. And in building a drug company, we didn't build this to be a single shot, one shot, and we're done. If you fail, you're done company.
Sure.
We built this based on the fundamental work that Mark and his team did at Temple. And by the way, 2 PhDs have been earned in his lab for elucidating all the mechanisms in the proteomics, and that research is continuing in the lab. So we think we're onto something pretty exciting that has branches into oncology, branches into immunology. And it's a question now of, can we prove this in humans in the clinic? And that, so we chose psoriasis as our beachhead, as our gateway. And we're very excited about the results we're seeing, but we have big ambitions for how to use this technology in other disease.
Mm-hmm. Yeah, particularly like I saw something in there about the cytokine release syndrome, which is, such obviously a hot topic because of COVID, but at the same time with the CAR T therapies and everything happening in the Philly area on those immunotherapies, the ability to modulate that whole part of the immune system while having the oncology of benefits is extremely important. So I was excited to see that you were working there as well. Ira, what's coming up? I know that you guys are doing a fundraising for the phase 2 development. Anything else as we are, sort of getting further into 2024 now that we should know about in terms of that program, other initiatives that are coming up in terms of public-facing stuff for the company? Conferences, talks you're going to be giving, please. Anything else that's you want to highlight, please?
Well, so Jim Kerwin, our Chief Operating Officer, who's a very experienced clinical trialist, who was my deputy and right hand for over 5 years at Wyeth, is going to be presenting at Bio New Jersey, JP Morgan East
Nice.
in May. We have been working on a publication strategy, and we're shifting gears from being very focused in the lab and very focused on patents to try now to get the word out about what we're doing. So there will be more coming. We've had a chief medical officer, Stefan Weiss, who's an eminent dermatologist, just join us. There'll be a press release coming out in a couple of weeks on that. We will be raising a Series A later in the year, driven on 2 or 3 events. The first is that we're going to be filing the phase 2 protocol design with the FDA in the coming months. We're going to be publishing the phase 1B second cohort results this summer, and then pooling the cohort 1 and cohort 2 and publishing the complete phase 1B trial results, which will be on 30 subjects. The cohort 1, by the way, results were spectacular. They were world class. And so cohort 2 is a variation of that drug, which we think will be faster acting. It has an additional adjuvant added to the drug that we think will improve onset of action. One of the things that's different about what we're doing in modulating cytokines, and particularly, we hit TNF alpha, IL-23, IL-17, IL-12, and interferon gamma, which is, by the way, an identified target that's not been drugged before. We also block the upregulation we upregulate IL-10 to block autoimmunity, which is a completely new pathway, and that's patent's been filed. So there's a lot of interesting work going on here, and there will be more publications coming out in the coming months. So it's both a laser-focused approach on psoriasis, because that's what we need to prove that SFA-002 really works, and that to raise the money we need for the Series A. But then behind it, there's this platform with drug after drug after drug. And we're using short chain fatty acids as a palette. And we've learned how to manipulate their effect on specific cytokines using adjuvants to target specific diseases. So we're not done yet, is what I'm saying. And this was not built to be, a single shot company. This was built to be something that can change medicine. And we're pretty bold about it. I know it sounds audacious, but we think this is an entire class of molecules that have been largely overlooked because people said, well, I remember presenting this in September of 2017, and you could have heard a pin drop after the presentation. There were no questions. There was nobody interested. People said, "Oh, it's natural product derived. You can't patent it." Well, we have 10 issued, 4 allowed, and we're on track for 20 patents by year end. So yes, you can patent it. Yep. People have said, well, the half-life is so short, you can't turn this into a drug. Well, yes, we've actually shown that we can create sustained blood levels, 24 hours, and we figured out a pathway to do that. The toxicity is very clean. The FDA has been very supportive and cooperative. We actually have a good relationship with the agency. So we think we're onto something very cool. That's why I'm still doing this. I mean, this is going to be my 35th drug, and I think it's something cool.
It's extremely cool. Ira, say a few words about the upcoming round that's heading up in the fall.
Capital is fuel for small biotechs. I mean, and my job as a CEO in part is to continue to raise the funds we need to fund our development work. Driven by data, the outcome of the phase 1b trial, the filing of the phase 2 protocol, and hopefully the approval of the next phase 2 program by the FDA. Our plan is to raise between $25 and $40 million in a Series A raise in this fall. We previously raised a bridge of $4.5 million led by North South Ventures. We have a lead for the Series A raise, and that's going to be very important for funding the phase 2 program for SFA 002 in psoriasis in the next 2 years.
Excellent. It's novel, it's natural product, it's multi-mechanism, you got a platform, and it's Philly. So I mean, I love it all. I'm rooting you on. I can't wait to see what happens next, both on your phase 2 front, obviously your fundraising, and the future development of the company, and it's happening in the local bio ecosystem. So I'm excited for you, wishing you and the team the best with it as you continue down the path. Again, for everybody that's going to be listening to this particular episode of our show across the various podcasts, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Ira Spector, co-founder SFA Therapeutics. We will be putting a link in the bio of the show. Ira, I really want to thank you for taking the time out of your schedule to come tell the story about everything you're up to. Obviously, thank you for what you're involved in, both on the inflammatory and the oncology front. And as we like to say on our show here, thanks for helping to create a better tomorrow via what you're doing to develop these novel platform technologies. It's a really great story, and wish you and the team the best with it.
Thank you very much.